In this video, we're going to be looking at proteins. So let's start with amino acids. This is the general structure of an amino acid. We have a central carbon. On one side, we have an amino group, NH2, and on the other side, we have a carboxyl group, which is also found in carboxylic acids, hence the name amino acids. There's always a hydrogen attached to the central carbon, and we have a variable side chain. Now, there are 20 different versions of this variable side chain, which is why there are 20 different amino acids. These side chains are responsible for giving the amino acid unique properties. Okay, so amino acids can join together, and that is thanks to the following groups on the amino and the carboxyl group. So let's see that in action. So here we have two amino acids. We've circled the parts of the amino acids that are going to join together, and they condense together to release a molecule of water and create a dipeptide. This is known as the peptide bond. It can also be written as CONH. You can continue adding amino acids on either side of the dipeptide to make the chain even longer. So, a chain of many amino acids joined together is also known as a polypeptide. Now, let's say this polypeptide has an initial structure of just a simple chain. A protein, however, is much more complicated, and this is what the final structure is most likely going to look like. So, let's see how this simple chain of amino acids ends up looking like this complex structure. The primary structure of a protein is simply a sequence of amino acids that are joined together by peptide bonds. By the way, this is the most important part because any changes to the primary structure will cause the final structure to also change. Okay, so let's represent that structure as a simple line. Soon, parts of this will coil up and fold like the following. This gives us the secondary structure. A coil is known as an alpha helix, and the folds are called beta sheets. And this is because amino acids have NH and OH bonds. This is a tertiary structure, also known as the final 3D structure of the protein. In addition to hydrogen bonds, we also have ionic bonds and disulfide bridges. Also, amino acids which are hydrophobic, meaning hate water, will tend to move towards the inside of the protein, and those that love water or are hydrophilic will move towards the surface. But we're not done yet. What would happen if I take 1, 2, 3 proteins and join them together? This will create a superprotein, also known as the fourth and quaternary structure. Now, the quaternary structure is defined as 2 or more polypeptides joined together. So let's look at disulfide bridges in a little bit more detail. In this chain of amino acids, we can see that some amino acids have sulfur in their R groups. The ones with sulfur are known as cysteine. When the two sulfur atoms are close together, they will join together and create a disulfide bridge. This causes the polypeptide to change shape. Let's talk about how we can test for proteins. We add sodium hydroxide and burette reagent, which is copper sulfate, to our test tube, and if proteins are present, it will change color from blue to purple. So here's a good way that you can remember that. B for burette, B for blue, and P for protein, P for purple. Remember, if there are no proteins present, it will simply stay blue. Hey guys, if that video helped you, support our channel by liking, subscribing, and sharing it with your friends. And more importantly, if you still have questions, drop a post on our forum at ExamQA.com, where I will personally be there to help answer your questions. Mohammed signing out.